All right, let's get started, guys. Did anyone see the Nobel Prize winners this year? Anything weird about it? No idea? Okay, let's start by the medicine. So the medicine Nobel Prize was won by Ambrose and Rufkin. They were it was shared for the discovery of microRNA. So this is +1 for genomics. MicroRNA, if you remember, I talked about them in the beginning of the course. This is basically one of the functional RNAs that are found in the genome. So this makes sense because microRNAs have a lot of functions in disease and all that. So that's one thing. The weird thing is here in chemistry. Anybody ever seen this name here? No? Okay. So Demis Hassabis is the previous CEO of Google DeepMind. And this is the new CEO of Google DeepMind. So these guys have no chemistry background whatsoever. But they won the Nobel Prize for Chemistry because they contributed the software that is called AlphaFold. Did you ever hear about AlphaFold? You did? Okay. So AlphaFold is, let me just do it like this. So AlphaFold is a software that was created using AI, and it allows you to, well, more like takes advantage of AI technology to predict the 3D structure of a protein. Why is this such a great feature? It is a great feature because predicting the structure of a protein is quite hard because of the different conformations and interactions between all the atoms and all that. And so before, the way you would do it is by using X-ray crystallography, and you would do that, and sometimes you don't get the full thing, etc. So this here just takes the sequence and then gives you the 3D structure. Okay. So the only chemist in this team is this guy. The other two are not chemists. They're CEOs of previous CEOs and current CEO of Google DeepMind. So if you remember the AlphaGo, the AI that beat the Go, the Pro in Go, whatever, which is a board game. Okay, so these are the guys behind it. They have no background in chemistry, but they use AI to help chemistry. So I think this is one of the first, if not the first, prize that was given to a non-chemist, but for chemistry. Weird stuff happening. It also seems like the Nobel Prize is changing their preference. Where was it? Jeez, 1 sec. So, God, what am I doing? Give me 1 second. So this year there's a few winners. So the physics prize was also given to researchers that worked on the machine learning and artificial neural networks. So there's 2 prizes that are linked to AI. So I think the Nobel Prize Committee is starting to like AI in a way. But yeah, this is quite interesting. This guy is from Toronto. Hopfield is from Toronto, yeah. Cool. So get into AI ASAP. That's what I have to say. Okay, so today's lecture. Let's see. Today's lecture. Before I get into the lecture, do you guys have any questions for me? No? Okay. So the assignment that was due on Friday is still due on Friday. The essay has been moved to Sunday, 11:59 p.m. This way it gives you 2 whole days during the weekend to work on it. Pretty sure it's not going to change much, but you know, if you have a busy week, maybe it's going to help you. Okay. Shauna, I got your email. I will reply to it in the next few hours. Alex, I also got your email. Yes, your paper is good. It was Julian and who else? Yes. Okay. Cool. What's your name? Okay. Cool. So let's get started by today's lecture. Okay. So today's lecture is going to talk about the genome assembly, the state of it right now, and then we'll get into a little bit of annotation. And that's pretty much it. If you have questions for me today, let's try to either send me an email or meet up next class or something like that, because I have to leave quite fast after the lecture. I have to go pick up some stuff. So today, no questions after class, okay? So today we're going to talk about the timeline to get to the genome assembly that we have right now. So the genome assembly obviously is not something static because there's always updates that happen to it. Yes, of course, we have the complete genome, but we don't have all the variants at all the nucleotides in the DNA. And obviously, we'll talk about the type of questions that get asked after you sequence the genome, because initially it's more like the question is, what is the genome? What is it made of? What's the sequence? And then we start asking more complex questions. For example, what does each part of the genome do? And how does it differ between each person? So this is what we'll take a look at. So just a little bit of history. In 2001, if you remember, I told you that the draft sequence was published. There's a Nature and a Science paper on that. And that was not really the complete genome. It's called the draft sequence, which comprises 90% of euchromatin. Euchromatin is the gene-rich portion of chromatin. So this is not all the genome. 2003, we talk about the complete sequence. Obviously a consensus sequence because it varies between people. So that was about 99% of the euchromatin that was sequenced. But there was an issue, and there were many gaps in this complete sequence due to repetitive sequences that were quite complex to place on the chromosomes and toxic sequences which could not even be cloned. Remember, at this time, we're still using cloned. We're not using direct sequencing on fragments. Okay. So the repetitive sequences were fixed by long read sequencing. So if you increase the read length of your sequencing, you will kind of have two parts of DNA that are on the side of a repetitive sequence, so you can kind of get the whole repetitive sequence, and whatever is on each side can help you map. So that's that. Toxic sequences, it was solved by having next-gen sequencing, which is basically just sequencing directly from the fragment. This way you don't have to clone it, and if you don't clone it, you don't kill whatever bacteria or yeast you're using. In 2010, we thought we had 357 gaps in the genome that were still not completed. But after some time, we actually realized that there were 819. These gaps were on chromosome 1, 14, 22, and many others. These ones had about 8 to 9 gaps. So you can see that as you increase the genome coverage depth, you can never really reach 100% of the genome unless you resequence and you use different parameters. Okay. There's a consortium called the Genome. I forgot let's take a look at it. I always forget what it is. I think it's Research Consortium. Okay. So these guys here, what they do is that they basically give you the information on the genome, and they take care of curating and making sure that the assemblies are done properly and all that. So they take care of all this, okay? And they make updates to the genome assembly. So the genome assembly is, what the hell is happening? The genome assembly is basically each time you sequence the genome, you will assemble it using the contigs, scaffolds, and all that. And that will give you a certain assembly. But it is not always the same depending on which team is doing it and depending on which method is used and which workflow, right? So you end up having multiple assemblies done by multiple research groups in the world, and then you have to kind of put everything together, okay? So this Genome Research Consortium created an assembly that is the same for everyone, and currently they are at this is H for human, and 38 is the assembly iteration. So they started with 1, now we're at 38. 1 was the 2001 basically, okay? And then they keep adding patches to this. So every time they will add a patch, just like you were adding an update to a software, and they're adding either missing sequence or they are correcting inconsistencies in the genome after resequencing multiple times, okay? For example, there are a few genes that are quite variable. For example, the major histocompatibility complex. This is the complex that tells you if your cells are your own or not. So when you do a transplant, this is what gets activated. This is what gets detected and then detected as foreign kind of organism or whatever. And so obviously you expect a lot of variability in it. And because there's a lot of variability in it, then you end up having variation in your assembly. So in the case of this assembly here, they have about 261 alternative sequences for just MHC and the LRC, which basically have similar jobs. Okay. Obviously, it's not perfectly like from end to end, you know the sequence, and it's the same for everyone. There are regions that are highly conserved that have the same sequence for almost everyone, and then others like this MHC complex, which are totally different between people. At this point here in 2014, there were 42 contigs that have been not placed. So we know the chromosome, but at the base pair resolution, we don't know where to place them. And that's just because of the repeats, right? So the repeats are kind of repetitive sequences, and because they're repetitive sequences, it's like, where do you place it? If you have 3 repeats, do you place it at the first repeat, second repeat, or third repeat, right? So that's that. And then you have also 127 that we don't know where the hell they go because we don't have any sequence that is overlapping with others. Okay, how does this happen? Obviously from the fragmentation pattern that you get and your genome coverage. 2018, they had the P13 patch. I think right now they're at, let me check where they are. GRC 38.P. They're at 14, okay? So they're at patch 14. Basically the 38, every patch they add, they will just make improvements to the assembly by correcting different problems. They were expecting PacBio and Oxford Nanopore, which are long read sequencing methods. As you remember, it does like 100K or something like this in one go to fix the gaps, but obviously this is not sufficient. You need to shift your start point and then keep sequencing. There was a plan for GRC 39, so the 39th iteration of this assembly, but they're still working on figuring out what's the best way to have the coordinates for each of the nucleotide. So until then, they pushed basically the dates to indefinite until they figure out what's the best way to map the DNA. So I'll just show you something here so you understand where does this UCSC browser. So you understand what this means. So the genome is found here. You have the GRCH37 and 38. So you can take a look at whichever you want here. If you click on it, you will see that this is the assembly that you have. Obviously, this looks very weird. Let me just zoom out. And you can see all the genes that are found. And obviously, there are still gaps to be found. I don't have one specific here, but you can for sure look in here and you'll find gaps. So there's a coordinate system for each of the nucleotide that changes every time they iterate this system. A problem with this is that when you're doing research, you end up having to convert your current mapping coordinates to new ones, and that leads to a lot of problems, but obviously you end up with a better data set. So you can see all the genes where they are, okay? You can play with this. For example, if you want to find the BRCA gene for breast cancer, you can just write it here. And if you zoom out, you'll see that it is on chromosome 17, and here is the starting point, and here is where it ends. Okay, there it is. Cool. Why is this useful? Depends if you're doing research or not. You'll be using this a lot, okay? So we have many assemblies. Now, how do we make sure that the assembly is quite good and that is, geez. Should I go outside then? I feel like this is the time to do it. So the assembly statistics, you have to make sure that your assembly is good. And so there are a few statistics that were created by researchers and bioinformaticians to make sure that this data is right. Let's start by the two different assemblies or groups that take care of this. So you have the RefSeq group, which is kind of highly curated data of the genome, data set of the genome. They verify every sequence, they verify everything basically. And then GenBank is less curated, but it allows for data from researchers like you and me. And this is important because in this case here, you end up with identifying variants, because these guys are not people that publish here. They get the DNA from many people. And so you end up having more variants, while here they try to find the least redundant and most stable assembly of the genome. Well, in this case, it's free for all, kind of. And so you could go and put your own sequencing here, and you'll have obviously the variants and all that. They're pretty similar. The genome size is 3.1 billion bases. There's 2.9 billion that have ungapped length. So this means if you take just the DNA that has no gaps, so we're pretty close, but there's about 200 million that are missing. How many gaps there are between the scaffolds? So if you remember the scaffolds, when you have multiple contigs, you put them all together. So that makes you a big contig like this. And then if you sequence from both sides, you'll end up having the sequence here and here. And so scaffolds are just combinations of these multi-contig units, okay? So there's 349 gaps, which is the same as GenBank, so they're quite close to each other. The number of scaffolds, there's 3 scaffolds that are different here, probably originating from DNA variant or from DNA that was not found here. The N50, L50, and all that, we'll talk about it. Here you have the number of contigs, which is a difference of 3. So these guys seem to have 3 more contigs in their data set. Again, more statistics, which we'll talk about, and that's pretty much it. If you take a look at the genome assembly, I'm just presenting this to you, but honestly, it's just to show you what's happening right now, but it has no meaning for actually the theory or the stuff that we're learning. It's just to show you the state of the assembly that we're in right now. So there's all these chromosomes. Don't forget the mitochondrial DNA. And then you can see the size of the chromosomes. Chromosome 1 is the biggest one. There is also the number of ribosomal RNA, tRNA, and all that in these chromosomes, which were identified. And these will be interesting when we'll talk about the functional RNAs in the course. We have the number of genes per chromosome also, and pseudogenes, which we're not clear about. One question for you guys. Which chromosome here was the hardest to sequence? Pop quiz.
Hardest as in took the longest time?
Yeah.
As in was the...
Took the longest time, yeah.
But that would be number 1 because it has the biggest size.
That's what people would think, but no. Anybody? Sorry? The smallest one? Why the smallest one though? Or just like this? Okay, it's the Y chromosome. The Y chromosome is the smallest chromosome, and it was the hardest to assemble.
Because half of the people have it on, right?
Actually, that's possible, but the reality is that the Y chromosome has so much repetitive sequence in it, and has a lot of sequence that is hard to place when you break it into contigs and all that. So the Y chromosome is kind of messed up. It was the last one to be sequenced, and it was due to long read sequencing. Otherwise, you can't do it in the other way, okay? Which is kind of weird. Forgot your name. Heidi. I will remember. Okay, Julian, Heidi. Weird, huh? Seems like the biggest one should be the one, but nope, apparently not. Julian, and then Heidi. Okay, more statistics. What was the point of this? I put this here just for this part here. So 35,000 clones were used to sequence the human genome. Okay, so you can imagine how many 384 or 84 well plate were used for this. It's a lot. Okay. and the number of contigs 998. This is the latest statistics. Okay. What else? What else? So in here, I'm just showing you that there's a lot of the DNA that is still not. We say we have a complete sequence, but not 100%. There's still a lot of scaffolds that are unlocalized. For example, here, chromosome 1, there's still 742,000 bases that are, we don't know where the hell to put them. Okay, so this seems like not a lot, but this is at least 20 genes, 30 genes, something like this. Okay, so there's that. So I've been talking about multiple assemblies. Now, how do you make sure that an assembly is actually good or not? So there's a few statistics, or more like measures of quality of the assembly that have been created. There's the N50, L50, and NG50. And these are designed to kind of capture if the assembly is complete or not. For N50, the bigger the better. For L50, the smaller the better. And NG50 goes with N50, okay? In the same direction. So let's start by N50. So N50 takes a look at the contigs and tells you basically the quality of the genome based on the structure of the contigs. So the way it works is that you'll take all your contigs and order them sequentially. So contig 1, 2, 3, 4, 5, 6. And then you order them from the largest to smallest, and you go basically until you find 50% of the total assembly size. So if, for example, your assembly, you have 800,000 nucleotides, 50% is 400,000, okay? In this case, in this example here, the full length of the assembly, not the genome, the assembly. So these are two different things, because the assembly could not be fully representative of the genome. It could be part of the genome. And so because it is part of the genome, you need to have some sort of measures to make sure that it applies to the full genome. And these are the ones that we are looking at. So in the case of this assembly, which has 400,000 bases, then if you place your contigs in terms of size in a sequence, from largest to smallest, you will count until you reach the 50% mark. So at 200,000 bases, that's where you stop. Now, how do you determine your N50? Basically, you will count all the contigs that you had to go through until you reach 50% of the assembly. So 100 plus 70, that's 170. So that's still not 50%. Plus 60, now we're at 230,000 bases. So your N50 is this fragment here, okay? So then you consider your N50 as being 60,000 bases. Now, why are we thinking of it like this? Because the bigger your contigs, this is basically your median, okay? The bigger your contigs are, the better in terms of quality of your assembly, because if you have a genome this big and you divide it into 3 pieces, if they're all big, then you end up being sure that there's only, few variations where you can kind of flip them around. But if you have millions of contigs or thousands of contigs, if they're all small, it increases the risk of misplacing a contig in the DNA, right? And so as you count the contigs until you reach your 50% mark, the bigger this number is, the better, because that means most of the contigs in your assembly are huge, okay? And they're not small. Yeah, makes sense? No?
Isn't it kind of a flawed metric?
Tell me.
If you take the median piece, but like the lower half is full of like 200 individual ones, it's not going to tell you.
It is indeed. It's not the perfect statistic. But I don't see. I almost call this statistic, but it's more like a measure. It's not the perfect measure, but this is kind of a approximate way that it's being done. Because if all this here is bad, is like super small, then obviously you are right, indeed. But apparently this is what they're using for this. What would be a better way to do this? Average size of your contig, for example? That would be good, but I don't know why they're using this N50. I just remembered your question. I Googled it. I will tell you about it in a few seconds. So So this is one metric. The other metric is the L50. L50 is the minimum number of contigs to get half of the genome length. So the less contigs that you have, the better. This makes sense, right? Because they are bigger, and because they are bigger, so these two metrics are based on the size of the contigs. The bigger they are, the better, okay? And so if you have bigger contigs, you'll have a smaller number of contigs required to reach 50% of the genome length. So if we go here, the scaffold N50 is about 67 million bases for the human genome, and the scaffold L50, so the number of scaffolds required to reach 50% of the genome, is 16. Okay? If you can make this smaller, then it becomes much better. Okay? And then you have the equivalent for contigs, but just imagine scaffolds are multi-contig units. Okay? So this does not solve one problem, because if your assembly has 100,000 bases, and then you have a genome of 1 million bases because you measured the amount of DNA found in your nucleus or whatever, then your expected number of bases is 1 million, but you have only 800,000. So your L50 and N50 are measured in relationship to the 800,000 and not to the million. So NG50, what it does is that it just corrects the actual size to the actual size of the genome. Okay? And this allows you for comparing assemblies in absolute values. Okay, so just multiplications. What did I want to say here? Okay, I forgot. Maybe it's going to come back. So I showed you a paper that closed the gaps in the human genome. So there are a few of them that keep popping up to close the gaps in the human genome. In 2000, there's a team run by Miga and Corin and others that generated a gapless telomere to telomere assembly of the human chromosome X. And they did this by using this T-form mole cell line because these cell lines are haploid, and so there is no issue about the different alleles. Okay, you'll see if you go on the browser here, you'll see the T2TCH. Okay, so now you know what the hell this means. Make sense? Anybody know what T-form mole means? Okay, some embryology class. Let me tell you about this. So there's what happens is that when there's fertilization, sometimes the egg does not have DNA, and because the egg does not have DNA, the sperm that goes in basically is the only provider of genetic material. And because of that, you end up with a haploid cell that is called a hydatiform mole, and that continues dividing but does not make an embryo. What's interesting about these cells is that they have one copy of each chromosome. So you end up with the X and the Y in the cell, and they're very interesting for studying the human genome, because they provide you for a copy of each chromosome in a haploid state. Okay? So this is what it is. So this is why this cell was chosen to do this kind of work. Their work took the X chromosome, which had 29 gaps compared to the reference sequence, and they basically fixed all this, and they used next-gen sequencing. Okay, I think it was nanopore, but I don't remember 100%. 2020. What else? The first autosome was also assembled. T2T means telomere to telomere, and this was chromosome number 8. And they used a combination of different methods. So again, as I told you, the first time you sequence is not perfect. You have to complement it with other data sets so you can be more sure about your data. So they used nanopore, PacBio, and 2 variations of PacBio. And they produced a almost 100% accurate assembly of the chromosome 8. Then 2021, we got all the 22 autosomes. So if you look at the progress, we started in 2001, and then we started before, but let's assume from the draft sequence. It took 20 years after the announcement to actually get the full 22 autosomes. The thing is, it's not even 100%. So we say complete. It's not really complete, okay? There's always holes in it. One thing that's weird about this also is that as time goes, the reference genome changes. And so it's kind of a dynamic process because you sequence, you get the complete sequence, but as you sequence, let's say in 100 years, it will not be the same reference sequence because you'll have some deletions, some translocations, things like this. So this will never, in my opinion, be complete, okay? For this project here in 2021, they used 30x coverage, PacBio, HiFi, 120 Oxford Nanopore, and all the different techniques. They combined them together. This led to 200 million bases of novel sequence from the draft sequence. This is a lot. They found 2,200 paralogous gene copies. So these are gene copies that are related to each other, and then either duplicates or whatever. And then of those, 115 are predicted to be protein coding. So imagine missing 115 protein coding genes. These are important for disease and all that. So obviously they are predicted because we'll talk about how you can predict if a gene leads to protein coding or not using computational methods. They still have to be tested and verified to make sure that these are actually protein coding genes. The last of the race, chromosome Y. So chromosome Y was sequenced just recently, last year, in 2023, published on the 23rd of August. This is weird. I think they did it on purpose. I don't know. Chromosome number 23, right? So there's that. And the chromosome Y was the hardest because of many things, including, where do they say it? Anyways, this is just the abstract of the paper, but basically, without reading it, I know they're talking about repeat sequence and all that. So you can take a look at this paper and see how it was done. This is just the funny part here about the 23, and the Y chromosome. So, was that clear? A little bit of history. Not really necessary for, it's interesting to know where the genomics field is, but obviously for us, it's not that important. I'm just telling you how we progressed, and obviously we're still continuing to do the sequencing. We're not done. So what do you do when you have the genome? What's actually the next step? You guys have an idea? Don't read the last one yet. Yeah, that's one thing. What else? Yes, Sarah. Yeah, we can do that too. Anybody else? Okay, so this part is clear for you guys. It's the first part that was okay. Let me just write this. What about resequence? We're not done. Yes, Maria. Okay, yeah, we can do some comparative genomics. That's the words. Maria, anybody else? That's it? Yes, Alessandra.
We can look for different alleles and different genomes.
Variants. Maria, okay. Yes, Matt.
Yeah, I was going to say you have the reference genome, but you need to know the spectrum of variation within all the sequence.
Yeah. Okay. So we're not doing that. We're first resequencing. Okay. We're not done with the sequencing. Once you have the reference sequence, obviously, yeah, you do have it, but it's not perfect. As you've seen, this process has been very hard to follow, or somewhat hard to follow. It's because it is not a perfect process. Okay. It's not clean 100%, and it's prone to a lot of errors. So what you have to do is you have to improve your sequencing strategies, your machines, and then you resequence until you get the full genome. But if you follow the Poisson distribution, you'll almost never get to it. Okay. theoretically. But still, you can get pretty close to it. And so now that you have the reference sequence, you can create primers without having to kind of cut pieces and do weird things with it, right? And so this way it facilitates resequencing, and then it makes sure to give you an additional confirmation of that specific part of the DNA. Instead of now having two contigs like this, now you can, for example, do long read sequencing and read from end to end. This will tell you if you missed something in the middle, or if you have, for example, your repeats, if you missed one repeating unit or 10 repeating units or things like this. So there's a lot of things to do. At this point, we have commercially available kits that you can use, as I showed you for nebula sequencing. It's a small kit that you take, send them your DNA, and they do the whole thing. And this was just answered, so you can actually compare the human genome you try to find differences between the genomes. So obviously we don't all look alike, and so we have differences in our genomes, and so you can find these variants and then understand what they do exactly. So in 2012, there was a project that was aiming to sequence 1,000 genomes. So this is at the time where sequencing was not that fast. It was quite fast, but not as fast as now, where you can do it in a few hours. The goal was to try to identify enough variation to identify basically the variant that exists in at least 1% of the population. This would give you kind of fairly accurate, but it's not really 100% accurate, variation of human genetics between people. And this is basically just re-sequencing the genome, okay? So they published 1,092 genomes in 2012. These guys are funny, so they aim to go 10 times more. So they went for 10,000 genomes in 2016, and this was done by Craig Venter and the Institute on Human Longevity. They used Illumina, which is the most accurate of the sequencing technologies, and they got 10,545 genomes to 30 to 40-fold coverage. This is pretty high. In this project, they were able to identify 150 million single nucleotide polymorphisms. This was not available. This was not possible before because you didn't have a lot of genomes to compare between people. So now you can see that there are places where there are these SNPs. And they found tens of thousands of variants that were not identified before for certain genes. They added about 700 kb sequence per genome. Yeah, because basically 700 kb of each of these genome, on average, each of these genome had 700 kb sequence that was different, and about 8.5K variants. Okay, so this is quite a lot. The approach with Illumina, so remember which one was the best for identifying the SNPs? The 2-base one. Which one is that? That was the solid system that was pretty good. Which other 2-base system was there? Electricity one, what is it called? Come on, guys. Nanopore. Remember, it gives an electric signal, and I told you that the signal comes from 2 nucleotides. So those are much better to finding the SNPs and all that. And so this is not the perfect method. Obviously, there's another group that came up and wants to do 100,000 genomes that are identified. Currently, so the idea was to sequence 100,000 British genomes, and I think there was a problem that happened here, but they are looking at 15,000 people with rare genetic diseases. So this is interesting because now they're targeting directly the variants, and 25,000 people with cancer. They went down a little bit because obviously to get this much genomes is quite hard. You can read on this here. And then obviously you get to the million, okay? So there's a 10x, just keep 10x everything. And this is a consortium that was created in Europe. Usually, I don't want to say this because for certain things it's true, for others not, but usually Europe is very good at collaborating in terms of research. They usually are the ones that create these consortiums that include multiple countries. Croatia was the last to join this group. Obviously, last week this does not apply. This is like from his slides, okay? So it's been like 2 years. But Croatia was the last country to join this group, and there's about, I think, 20 countries in here, maybe a bit less, that have joined this consortium, and they all provide genomes to make it available for sequencing first, and then to make it available for everyone. So you can find this online. Just Google it up. Okay. So that's it for this part. Now the annotation. So what is the structure of the actual genome? Because we have yeast, we know that yeast has genes, functional RNAs, telomeres, centromeres, and all that. But what about the human genome? Does it have the same structures? Obviously, in terms of length and size, it does not have the same size, right? It's much, much bigger. So does that mean, are we adding more functional components, more structural components, or is it just copies of the same ones, or what is happening, right? So the human genome is actually divided like this. We have obviously two genomes, the mitochondrial genome and the human genome. And mitochondrial is of bacterial origin. So you can see how most of it is actually protein coding, and some of it is regulatory RNA and sequences. And the 2%, less than 2%, is mostly repeats. As we increase the complexity for the human genome, you can see that actually the opposite happens. So we have only 1% of the genome that is protein coding, 45% that is transposon based. So this is weird. Why do we have 45% of transposon based? So these must have a certain function that was not here. And then obviously this is an additional level of complexity. This is non-protein coding. And so our initial idea about does the amount of DNA explain the complexity in terms of proteins? No, it does not. It's actually the non-coding DNA that is important for this. We have also 44% of other sequences. Other sequences means repetitive sequences and things like this, microsatellites, things like this. And then 4% of the genome is RNA genes and regulatory sequences like enhancers, silencers, and things of the sort. Okay. Don't look at the next slide. Which one has the largest genome? You're looking at the next slide, huh? Yes, it is amoeba. It is amoeba. So you would expect it not to be amoeba, but to be actually humans, because we're much more complex, at least biologically, than the amoeba. But the amoeba has 670,000 million bases. It's basically almost 1 trillion bases. We have 3 billion, so we're missing 997 billion to reach amoeba. No. We're missing 667. Wait, do the calculation, okay? I can't do it. But I think they're like 20-something thousand times more, have more DNA than us. Why the hell is it like this? Any ideas? We clearly have more protein, different proteins than them, a lot more.
Is it because their enzymes are simpler, so they need more of them to do basic tasks, and therefore need to encode many more genes?
You know what? This is actually a good theory. Yeah, I will look at this, but this is actually interesting. First time. Next year I'm going to ask an assignment on this. Yeah.
Or maybe it's related, but maybe they have to express certain enzyme or proteins in different conditions.
So we're still thinking about the proteins, and what about the junk DNA? Do they have more junk, less junk? Less junk? So this is all protein coding? It's probably mostly junk. probably. But that's a very good theory about the enzymes. You need more of them. Yeah.
But junk DNA, when we say junk DNA, is for nothing, or we don't know.
We don't know what it is, so we call it junk. We don't know what it does. But it does not code for proteins. That's the old way of calling it. I don't know if they changed this way of saying it. But junk DNA used to be referred to as DNA that basically had no function or no known function that you could technically remove and still have a viable organism. Okay. Let me just write people here. Yeah, I think that's it. Okay. Cool. So fruit fly, only 100 million, 180 million. Onion has 100 times more than that. The newt, which is a frog, has a lot, and then the amoeba, which is a crazy number. Obviously, we don't know what the hell is happening here because we did not sequence the amoeba. So maybe we should do that, right? So there's something called the genome complexity paradox. The C-value is considered the measure of genome complexity. Not really true because it's not really the actual complexity. It's more about the amount in grams of DNA per haploid. Okay, so amoeba has a lot more. Now you would think that a lot of DNA equals a complex genome, but actually this is not true because most of it could be useless or just repeats or redundant sequences or dubious ORFs that have no use. Okay, so the C-value is not proportional to the organismal complexity. It's actually usually the opposite. Do you guys have an idea why it's not linked to this complexity? Someone? No? Yeah.
Like you said, it could be just a lot of junk DNA.
Yeah. What else? Something new.
Probably just replicate stuff going on. It's just happening delivery systems that they don't do anything. They're just there.
What about introns? Introns, maybe introns, right? So, okay, so we'll take a look at this, obviously. I'll show you. Yep.
I don't know if it's possible, but maybe they don't have lots of ribosomes that they can work at the same time in the same sequences.
So they would need duplicates. Could be a good theory, but you would have to test, obviously. I'd be surprised, but yeah. So how much of the actual genome encodes for, how much of the genome encodes for proteins, not genes, okay? So you can see the genome size here of all the organisms, but if you were to take a look at this, you would think that this one does code for the most proteins. Actually, it's the opposite. So amoeba, we are not sure, but we know about the others. E. coli has the most genes per nucleotide, okay? Humans have the least. So it seems like this variation is due to the non-coding regions and not the coding regions, okay? Remember this slide? We're going back again, okay? So then how do you, knowing this, how do you define the components that make up the human genome? So we've seen how to do this in yeast, but not for the human genome. So we'll be taking a look at certain of them, not all of them, and then we'll be focusing on functional RNAs and selfish DNA, which are the two that we skipped kind of last time, right? We didn't go in depth in them. We'll talk a little bit about genes, but the rest not really, okay? So if you take a look at these different organisms, this is just an example to show you how the organisms are different. Human being the most complex in terms of biological complexity. E. coli being the least complex in terms of biological complexity. And so if you take a look at them, you can see that Saccharomyces cerevisiae, E. coli, Drosophila, which are pretty close to each other, are gene-rich, and they lack introns or don't have a lot of introns, and they have very little repeats. As you increase in terms of amount of DNA, that's found in the genome, then you end up creating these long regions of repeats, which we still don't know what the hell they do. Some of them, obviously, we figured out some, but still have a lot to do. And as you can see, in a 50 kb region for the maize, we have only one gene, while for E. coli, we have so many of them. So there's 1, 2, 3, 7, 8 genes. Okay, so you can see the differences here. Saccharomyces cerevisiae is pretty much similar to E. coli in terms of amount of genes per 50 kb. Humans is between the maize and the others, so it has some genes, a little bit more, but it also has a lot of repetitive sequence. If we take a look at amoeba, what would amoeba look like? This or this? Yeah. The maize, right? The amoeba would look more like the maize than the E. coli. And if you compare yourself to the maize, you can imagine that you're much more complex, right? So no need to explain that. But obviously, the sequence here is set to be junk because there's a little bit of kind of arrogance behind it. How come it has more DNA, but it's much more simpler than us, so it must be junk, right? But obviously not 100% true. Some of this sequence has function. The enhancers, silencers, and things like this are found in these regions, okay? They're not found inside the gene. Yes, Shana?
What exactly are pseudogenes?
Pseudogenes are considered genes kind of because they have the ORF, but they don't produce a protein. Right? So you think that they are genes, they have the start site and all that, but they don't produce protein. Fake genes. Okay, so what is a human gene? We kind of moved from yeast. Now we're dealing with a lot more stuff. So how do you figure out what is a gene? Is it a place that has an enhancer? Is it something that has a Tata box? Is it something that transcription factors bind? How do you figure this out structurally, right? There is another consortium called the ENCODE, which is kind of the continuation of the Human Genome Project, and their goal is to find the function of all the human genome. So all the genes, all the sequence, all that, okay? And their goal is just to annotate the genome to basically attach a function to it for each piece of the DNA. If you take a look, for example, here, I'll just show you for proteins, and then you can do some research. So if you go to UniProt and you write, for example, the gene that I am working on, let's do table. Okay, so you can see in different organisms, right? If you click on it, you can see that this is a reviewed gene. So that means it's a curated gene. It's not a predicted gene. That means it was done in the lab and all that. And if you go down here, you can find all the components, the molecular functions that were associated to it, etc. There's a lot of them that don't have a function associated to it. So there's still work being done to do that. And then obviously there's other people that will figure out where is it? All the different SNPs that happen, things like this. So this is a lot of data, but you can see these databases which are quite boring and hard to read, but there's people that are working on this. AlphaFold obviously is being used everywhere. That's why they got the Nobel Prize yesterday or 2 days ago, right? And then you can even get the structure of the gene and each of the domains. For example, here you know that there's a zinc finger, which is a DNA binding sequence. This is where the DNA binds. So if, for example, you want to play with this protein, you break this, then it does not bind to DNA anymore. Things like this, right? So if you're doing a research project, this information is quite useful. If you're doing synthetic biology work and you're trying to build the perfect enzyme, then you're going to go through this data set, find the enzyme that does whatever you're looking at, figure out where does it start and where does it end, cut it, paste it in another protein of interest, and then make your perfect enzyme from that, okay? So this is what these things do. Some of them are predicted by the computer. Others are curated, so you have to make sure you're looking at the right stuff, okay? Useless for our class, but I like to put, what we're doing in perspective so you guys see what the hell is happening instead of just me telling you these things exist, okay? So what does the gene what is the gene made up of structurally? So for humans, there's a lot of things. There's the introns, exons, promoter, terminator, the regulatory elements, repetitive sequences within the promoter, for example, Tata box, etc., transposons, and then you also have the functional annotation of this gene. So it's not just its structure, but you can also take a look at the molecular processes that it is involved in, the biological function. I just showed you this on UniProt, but there's many other databases. And so the work now is not just sequence and then function, but it's a lot more than that. We're looking at actually the specifics within the sequences. And it's a work in progress because there's a lot of sequence to go through. For example, if we wanted to take a look at the amoeba, I don't know how long it's going to be. Maybe I'll put this on the final. Yeah, the amoeba seems interesting, so maybe I'll do something about this in the final. Okay? So genes. Before, we used to do experimental methods to identify genes and understand what they do. Now we can also use computational methods, right? So there are two ways that we can use to identify the function of genes. So you could take a look at the sequence of the gene and determine its promoter, splicing sites, homology to other genes, etc., the whole list, based on just the computer. Because you know that the ATG site is the start site. You know that the Tata box usually is where things bind to start transcription. And so you can kind of do a blast to figure out all the genes that have a Tata box, right? And so this is a way for you to computationally annotate the genome by saying this is the promoter, this is the splicing sites, because the splicing sites also have specific sequences that are found in the gene. So splicing sites are found at each end of introns. And so you can then identify the introns and the exons inside of a gene. You can also compare it to known genes to see if part of a gene has a certain function. As I showed you the zinc finger of the protein that I'm working on, if you take it and blast it, you'll find all the proteins that have that zinc finger similar to it, and then you can say that these are DNA binding proteins. Now, what do they do? You can go on and do some experiments on that. So to identify promoters, you can use the Tata box as an example. And the Tata box is found in only 25% of promoters. So obviously, this is not sufficient to find all the promoters. There are variants of this Tata box depending on the gene, and these variants allow for the binding of different proteins. For the Tata box, there is a protein called TBP, Tata binding protein, which binds to it. But obviously, each variant has its own protein. So if you are looking at promoters in the genome, you can use this sequence, but you have also to take into account all the other variants to identify all the promoters. What's the goal of the Tata box? Should have asked you this as a question, but it's okay. If you remember from your classes, it allows for the binding of the RNA polymerase to make the transcript. And this is brought to the promoter by the TBP. Obviously, not all the Tata boxes represent the TAT3A sequences are not all Tata boxes, and not all genes have these Tata boxes. So you end up not covering everything with this. You need to look for the other variants. Okay, so this is one way that you can use to computationally find promoters. For splice sites, they have this sequence called the splice donor site and the splice acceptor site, and you have in between the branch site, and this is just where the cutting happens. So you can look at these sequences and find all the places where this occurs, right? Usually the splice site starts with, splice donor site starts with a GU, and then the splice acceptor site is AG. And so if you look at these, you can find introns where they start and where they end, okay? Another feature that is known is the 20 to 50 base between the branch site and the splice site. Not to get too much into detail about this, but it's just to tell you that sequences have a meaning, and so you can kind of figure out the pattern between all the introns, and you'll see that it's GU, AG at both ends, and then there's a branch site somewhere in there. So then you can identify all the introns like this. What else? You can also, what did I want to say here? The one problem with gene prediction is that obviously the introns don't all have this kind of sequence. Some of them are a bit different. So when you're trying to identify where genes start, where introns start and all that, it's a little bit harder compared to yeast, for example, because of the presence of a lot of introns and different splicing sites and different variants of the same protein. And so it's quite hard to identify all the different possibilities. So computers can help you doing this, but obviously it's not done for everything. So you can have a gene predicted as being this sequence from a start site to an end. So the start, the ATG, and then the terminator sequence. But then when you look at it more specifically, you'll end up finding that most of it is actually exons, but there are still introns in there, and so you have to filter out part of this, kind of curate this predicted gene to turn it into an actual gene. Okay? What else? The way you do this, you can take this sequence, turn it into mRNA. When you turn it into mRNA, then you have your intron-less sequence. Then you reverse transcribe it to make cDNA, and that will give you the DNA sequence, which then you can transpose on your predicted gene, and you'll find where the exons start and the introns start. Okay? There's a kind of a lot of tricks like this. It's not a one way and then you figure it out. Every specific gene has its own set of problems, but these are different ways that this is done. Okay? Now, obviously not all genes look alike, so this adds another level of complexity. If you start by going with the idea that there's a ATG and then a end tag site, then you can find a lot more than just 20,000 genes, because ATG or methionine is everywhere in the genome. That does not mean that it is a start site. And because of that, then you have to also add different ways to separate which methionines are found in the middle of the DNA of the protein, and which ones are actually the first amino acid of a protein, and which ones are at the beginning of the gene and at the end of the gene. So it's not only you find an ORF, which starts with ATG, and then you count 3 bases until you find TAG or whatever, one of the stop codons, and then you say this is a gene. That does not work like that, because there's so many combinations that work, but they're actually not genes. I just talked about the pseudo genes. These are, this is one way to find pseudo genes if you want to, okay? And so, yeah, so let me just remember what I'm seeing here. The interruption could be identified. Right, so this is just when you're comparing the genes between organisms. So let's say, for example, you have your enolase gene or whatever, hexokinase in yeast, and you want to find it in the human genome. So then you use that sequence, blast it, or align it with the human genome, and then you can find the sequence in the human genome, and then from there you can use that to figure out if the interruptions in your sequence that don't match are actually introns or not. Okay, so this is a way to find introns. There are a lot of tools that were created to do this. These you're probably aware of. You've probably been given a lot of exercises to do on this. The one I remember from my studies was in biochem 1. You had to use BLAST, but obviously I'm sure you had a lot of other classes. So you can use BLAST to compare different sequences between them. And then with these softwares, you can find different domains that are conserved between different organisms. You can find sequences like the signal peptides that tell you where they localize and things like this. You can find transmembrane domain across different proteins and things like this. But obviously, Whenever you do this computationally, it's not sufficient. You'll have to also make sure that it is true experimentally. So it's not 100% possible to just go through computational methods and 100% assume that it is actually, for example, a transmembrane domain. So with these methods, I think this is the last slide. So with these methods, you now are able to, or these softwares, you're now able to actually take a sequence of interest, run it through, let's say, BLAST, and it will tell you all the organisms that were sequenced where that piece of DNA seems to be appearing. And then from there, you can find the differences between them. You can, if you're comparing, for example, a gene from yeast, as I said, to humans, then you can find introns because yeast genes usually have a lot less introns. And so from there, you can compare both and say, okay, this is the hexokinase gene for humans, but it seems like there are parts of it that are not found in the yeast. So let's see if these are actually introns or if they are additional sequence that has a function in humans or not. And then you just do this for each gene until you have a structural annotation first. And then from there, you can go in the lab and test this experimentally, okay? There are many softwares that exist. BLAST-N is for nucleotides, BLAST-P is for protein. And then you can take a look at all this. And then obviously, some of them go forward and backwards. If you have the protein sequence, you can find the nucleotide sequence and things like this, okay? I don't think I have to cover this too much in depth because I know you guys use BLAST-N and BLAST-P for sure. So this should be it. Do you guys have any questions? This should be quite simple, the last part at least. So I would not be too scared about this. Anything? No? That's it? Okay. Yeah? Let's, when do you want? November? Well, you know, you can submit it the week, well, November is the presentations, let's start by that. November, the end of November is the presentations. So if you can prepare in 1 week and find me the article and, you know, go for it. But I suggest you do it a little bit earlier so that if you send me the paper today or tomorrow, I will give you an answer today or tomorrow. But then it's just based on you. Are you going to be able to prepare for the presentation? So don't give it to me like the week before your presentation because I know you're going to freak out. Okay, so good luck with the homework. I'll see you next class. And I know you have a lot of work to do, so go.